on development of nervous system. So this lecture includes the learning objectives. At the end of the session, the students should be able to describe the process of neurulation, the primary and secondary brain vesicles, nomenclature and the name of the adult derivatives, describe the development of cerebral hemispheres, along with it, the developmental anomalies of brain, skull, and meninges. So first of all, let's go back to the journal embryology, what you have taught in the human body module. So just quickly recall the definition of neurulation. If you remember, during the early phase, when we talk about the lectures in general embryology, so what was the definition of neurulation? This happens during third week of gestation, in which you find a dorsal ectoderm, one of the portion here, get neurified, get modified, and a tube-shaped structure is formed. This process is known as, or the formation of the neural tube from the ectoderm is known as neurulation. So first, as you see that close to the notochord, a deep concavity is appearing in the ectoderm, and then signals from the notochord or from the mesodermal cells start to make the layer shape structure after the completion of the gastrulation, and from here the inward folding, the ends of the neural groove fold or from the neural tube will fold to form the neural tube. This whole process is known as neurulation. Let's go to the formation of central nervous system. So as neurulation is started, this can be considered as the first step in the development of central nervous system. So when it started or happened, it is the third week of gestation. When you look the picture in the third week of gestation, you will see the shape of the ectoderm is like a slipper. Here you can find the neural plate, then the appearance of neural groove that will later on form the tube, and after the completion of the neurulation, these are the neural folds. And here are the somites that are the derivatives of development of the skeletal muscles that we have been discussing that has been discussed in the other module. And soon as you see, the lateral edges will going to fold to form the neural folds. Now let's discuss the formation of a neural tube. So as you see, this neural plate receives some signals from the underlying mesodermal cord. This is known as notochord. And from the notochord, signals reaches here on the ectoderm, and a groove or a cleft is appearing. This cleft is known as neural fold. And finally, it starts fusing in the midline to form the neural tube. So as slowly from cranial to the caudal direction as we go, we will see that fusion starts first in the cervical region, and then finally it comes to the cephalic at the lower part and the caudal directions. So what are the neuropores? These are the some terms that will come commonly when you talk about the development of nervous system, especially in general embryology. So neuropores, as you see here in the picture, first when the neural fold start appearing, and other surrounding structures you can see in the background like pericardial bulge that's related to the development of the heart, otic placodes that's going to the development of eye, and somites is for the skeletal muscles. You see first in the cervical region the fusion started in the midline. Slowly it is going to progress in the cephalic end, that is the head end, and caudal end both, as we discussed in the previous slide. As you see, it's coming down also and it's going up. So the terminal parts both at the cephalic end and at the caudal end remain opens, and these opening is known as neuropores. And this neuropore is going to communicate with the amniotic cavity. You can see the above This is known as cranial neuropore, and the below this is known as caudal neuropore. So now again, when the neuropore is going to close, as you see, this is the event of the third week of gestation. So close by the cranial neuropore somewhere close in the 25th day of the gestation. Different books, there was a little variation, but we usually took our references from the Langman's test book of embryology. So most of these days are taken directly from Langman's. And closure of caudal neuropore approximately took place 3 days later. So the first cranial and then caudal. So you can see that closure is happening in the 25th day for the cranial and 27th day for the caudal neuropore. Now we talk about the neurulation, we talk about the formation of neural folds, we talk about the neural groove. Now we are coming on the development of brain vesicles that can be considered as a precursor for the adult brain or the mature brain. Initially, you see now how the story is started. So you see the swellings appears in the neural tube. So first you see a swelling is appear close to the cranial region. So we can say that three brain vesicles or the three brain swellings appears in the cephalic neural tube. These are known as primary brain vesicles. So from cranial to caudal we can name them. First is prosencephalon, second one is mesencephalon or the future mesencephalon midbrain, and the last one is known as rhombencephalon. Or in the other word we can say that prosencephalon will be the forebrain, mesencephalon is a future midbrain, and rhombencephalon is a future hindbrain. Now when we look carefully, this prosencephalon will further divide it into two parts. The central part is going to develop, this part is going to develop in the diencephalon, and the lateral vesicles will develop in the telencephalon. Mesencephalon will continue as a midbrain, and rhombencephalon will again divide into two parts. One will be the metencephalon and other one will be diencephalon. Now, when these vesicles are appearing, you see the folds or the flexures are also appearing. We have two major flexures forming here in the brain vesicles. The cervical flexure is a fold that is appearing between the rhombencephalon and spinal cord, or we can say between hindbrain and spinal cord. The cephalic flexure is in the midbrain or the mesencephalon. Now we will take on later on what are the derivatives of metencephalon and diencephalon, and you can see in this diagram some cranial nerves are written here. So we can presume that from forebrain only the first two cranial nerves are arising. From midbrain, third and fourth, that is oculomotor and trochlear, and from 5th to 12th all the cranial nerves are arising from hindbrain. So let's go back in the details of the formation of brain vesicles. So this event started in the 5th week of gestation or the development, when they will be differentiated the primary vesicles into 5 secondary vesicles. So now you can see the most prominently telencephalon is appearing as a lateral swellings. The mid portion will be converting into diencephalon. The mesencephalon is the midbrain, and here one deep flexure is cephalic flexure that's appear in the midbrain. Now in between metencephalon and diencephalon, one more flexure is appearing here. This flexure is now known as pontine flexure. So Cranially is metencephalon and caudally is myelencephalon, and in between is a pontine flexure, as you can see. Now a deep furrow separates mesencephalon from metencephalon, and this is known as rhombocephalic isthmus. Here, this is rhombocephalic isthmus. This turn is known as pontine flexure. So this one is the future metencephalon, and this one is a myelencephalon. So pontine flexure marks the boundary between metencephalon and myelencephalon, as you can see here. And rhombocephalic isthmus is a separation between the midbrain here, that is mesencephalon, and developing metencephalon. So each of the secondary vesicles then contribute to the different parts of the brain. In extra, you can see the appearance of the cranial nerves. We'll talk about in detail later on. So prosencephalon, as we discussed before, consists of telencephalon that will becoming a future cerebral hemisphere. So from these lateral vesicles, the cerebral hemisphere is going to be formed. These lateral vesicles, this is the telencephalon vesicles that give rise to the cerebral hemisphere. And diencephalon is the midline portion from here, midline portion of the prosencephalon, from where the optic cup, pituitary stalk, thalamus, hypothalamus, and epiphysis is going to be developed. And the other part, as you see, the metencephalon and myelencephalon, the metencephalon will be converted into pons and cerebellum, and myelencephalon will convert it into medulla oblongata. Now we are coming in little bit details about the diencephalon. So as we know, the central part is going to form the diencephalon, or the central part of the prosencephalon is going to convert in diencephalon. And in this picture you see the roof plate of diencephalon, means the roof. It consists of a single layer of ependymal cells, and it is surrounded or covered by a vesicular mesenchyme. And what will happen later on, when the complete development took place, this roof plate will be converted into this structure. What is written here? It's a choroid plexus. And what will form by these choroid plexus? This will be becoming a secretory and absorptive function for the third ventricle. And the most caudal, that the tail end of this roof plate of diencephalon, will convert into pineal gland. You see here, this roof plate here is making a thick structure that will be the pineal gland or the pineal body along with the epiphysis. Now the alar plate, thalamus and hypothalamus. So now we are coming on the formation of thalamus, hypothalamus and alar plate. So what's the role of alar plate in the formation of thalamus and hypothalamus? So these alar plates form the lateral wall of diencephalon, and a groove known as hypothalamic sulcus, it's here in the picture. You see, this is a groove in the hypothalamic sulcus that divides this plate into dorsal and the ventral region. So this entire plate is known as alar plate that will be divided by hypothalamic sulcus. Now the dorsal part will be converted into thalamus and this ventral part will be converted into hypothalamus. And here in this picture or the transverse section of the prosencephalon, we can see the corpus striatum. This part is a bulge from the lateral ventricle, and this is a communication of the lateral ventricle to the third ventricle by a foramen. Now as we see, this thalamus gradually projects into the lumen of diencephalon, and both right and left side of the thalamus fuses in the midline by a connection that's known as in the adult brain interthalamic connexus. It's considered as a joint or a connection of the two sides of the thalamus. Now, cerebral hemisphere, as we talked about before, it's a derivative or the development of telencephalic vesicles, the lateral part. And when it arises in the 5th week of development from the prosencephalon. By the middle of the 2nd month, the basal part of the hemispheres, means the lower part, form the forward extension of the thalamus and begins to grow and bulge into the lumen of the lateral ventricles. To come to this picture or the diagram, you can see these are the walls of the telencephalic vesicles from where the cerebral hemisphere is developing. And inside these spaces or the cavity of telencephalon will convert it into the lateral ventricle. And below these are the fibers that are arising from here the cerebral cortex and passing to the corpus striatum here. And this is a third ventricle that's lying in the diencephalon. So this is now a cross-sectional complete picture showing initially the neural tube, then telencephalic vesicles, then the appearance of the cavities. These are the lateral ventricles, and this are the inner lining known as choroid fissure, as you see in the previous slide. And now this is a complete shape of cerebral hemisphere along with the lateral ventricle. So still we are considering in the cerebral hemispheres, the development of cerebral hemisphere. It's a growth going in the anterior dorsal inferior direction, and now it's converting into the lobes, frontal, temporal, and occipital lobe. The anterior one will be converting into the frontal lobe. The posterior one will be converting into occipital lobe. This lateral one will be converting into temporal lobe, and areas between the lobes, especially between the frontal and the temporal lobe, will appearing like a depression, and this big depression between the frontal and temporal lobe is known as insula. Now during the final part, as we see from 7 to 9 months or starting from the 5th month to the 9th month, what the change we can find in this picture. Let's see here the 5th month. Did you notice any sort of irregular depression that we know sulci and gyri? As we move up, this appears as a relatively smooth area. As we go in the 7th month, we see much more depressions and invaginations is happening. By the time of the 9th month, it completely encompassed or covered by these sulci and gyri. So this happens between 7 to 9 months. As cerebral hemisphere grows, the major part of it will get a convolutions that are known as gyri, and they are separated by the, and this convolutions will appearing as a sulci and gyri in the adult brain. Now, what is Commissures formation and what are Commissures? When you look at the adult central nervous system, you will find there is an innumerable number of the fibers or the bundles which crosses from one side to the other side. And these connections between the connecting the right and the left half of the cerebral hemispheres. So starting with the first commissure, commissure usually means the connecting fibers in the central nervous system. So what is lamina terminalis? Lamina terminalis is a crossing bundle of anterior commissure. First of all, the crossing bundles to be appear here is the anterior commissures. You can see here in the lamina terminalis, this is the anterior commissure. So what it consists of? It consists of the fibers connecting the olfactory bulb. Here below you have the olfactory bulb, this one, olfactory bulb, and the other related areas of the brain to the opposite side. So whenever the lamina terminalis, this layer is gone, inside it, this is anterior commissure, and it is connecting the olfactory bulb from one end to the other end, or the right to the left. Now, corpus callosum. What is corpus callosum? This is one of the biggest connections that is connecting the fibers from the right cerebral cortex to the left, and it appears in the 10th week of gestation, and rest of the non-olfactory area, that means the area other than this olfactory bulb, is connected by this big fiber band that's shown here by the dotted line. I can draw a red line here. This is corpus callosum, as written here, the future expansion of corpus callosum. So we talk about just a little bit, 1 minute here. We talk about the olfactory bulb, we talk about anterior commissure connecting the olfactory bulb, we talk about corpus callosum, and at the end you see the two small commissures that will be discussed. The details will be discussed in the coming module, but this development habenular commissure and this one is a posterior commissure. So these are all the connections. So let's summarize our lecture. We start with the definitions and the process of neurulation. Formation of the neural tube is known as neurulation, and how the neural fold is appearing and how the notochord is taking a role in giving a secondary signals to the formation of this neural groove and neural folds. Finally, these neural folds rearrange or appears in the form of the three vesicles, two flexures. Later on, these three vesicles will convert it into five vesicles or five divisions, and it will give to the future subcomponents of the brain or the central nervous system. Thalamus and hypothalamus, we talk about in detail a little bit about the development, how the alar plate is divided into dorsal part in thalamus and the ventral part is hypothalamus. Then telencephalic vesicles will convert it into cerebral hemispheres. And the last part we talk about is the formation of the commissure. This is all about in the development of nervous system part 1.